the first installment of our webinar series. We try to shed a light on the importance and role of oxygen in cell biology. My name is Daniel Danielsson with the Swedish pronunciation, and I work as a product specialist here at Milmedtek AB. We are incredibly thrilled to be hosting this event for all of you, and we're doing so together with Baker Ruskinn and together with Professor Randall Johnson from KI. So we're super excited about that. The lecture you're all attending is called Oxygen in Cell Biology, Episode 1, Oxygen and the Immune Response. Before Randall gets into his lecture and presentation, first me and Krista Rantanen, Director of Scientific Applications at Baker Ruskinn, will just take a few minutes to present the agenda and present our respective companies. So here is the agenda for today. We'll start off with this short introduction and then a presentation of my company, Milmedtek AB, here in Sweden. Then I'll hand over the torch to Krista Rantanen, Director of Scientific Applications at Baker Ruskinn. And then she in turn will hand over the torch to the main event, that is Professor Randall Johnson from Karolinska. We'll end the event with a live Q&A session where we'll be able to bring questions up on the screen. And then finally, for those interested, we will announce the next lecturer in this webinar series as well. Just a few words about the Q&A function. As you see in the chat pod in the lower right corner, Q&A has now been enabled. That means that you are able to write your questions throughout the webinar, and we will select a few questions at the end of Randall's lecture and bring up to the front screen. Please note that when we bring up the question to the front screen, we also get the name of the person who asked said question. If you want to remain anonymous, you can write anonymous before a question, and then I can write the question for you. Why I'm saying this is because this webinar is being recorded, and if you don't want your name to show up on that recording, then we can take the anonymous route. All right then, just a few words about Milmedtek. Our slogan, translated to English, is Tailored Laboratory and Industry Technology, and that's sort of what we're striving to do. We try to find tailored solutions for our customers when it comes to technology. We're located in the south of Sweden, outside Karlskrona, and we supply, install, and maintain both simple, everyday laboratory equipment to more advanced systems, such as Baker Ruskinn's Hypoxia chambers. We're the general agent to several leading manufacturers worldwide, including, of course, Baker Ruskinn, who's hosting this event together with us, Coyer, who produce top-of-the-line microbiological safety cabinets, Fedigari, who produces tailored and customized sterilization solutions to industry and labs, and Etaluma, who has incredible inverted fluorescence microscopes that can allow you to do time-lapse studies and much, much more. If you have any inquiries about laboratory equipment in Sweden, such as these, of course, these Hypoxia and anaerobic workstations from Baker Ruskinn, feel free to visit our website, www.milmedtek.se. So that's just a short presentation of us. I'll now hand over the torch to Krista Rantanen at Baker Ruskinn.
Thank you, Daniel. Thank you. And thank you all from my side and Baker Ruskinn's side as well. You're joining us today and tuning in to think about oxygen. So before I hand over to our esteemed speaker, I of course want to introduce Baker Ruskinn to you very briefly. So Baker Ruskinn is based in UK, and it's part of the bigger global Baker company that's based in Maine. And let me take the slides that come with these. There we go. Okay. there you see them. So Baker is the mothership over there in Maine, and Baker Ruskinn then in UK, and Clean Air by Baker in the Netherlands. So we are the world leaders in contamination control and precision atmospheres for life science industry and research. But we're not only the world leaders, we were also the first ones to develop these products. So Baker actually developed the first biological safety cabinet in the 40s, then followed by Baker Ruskinn developing the first hypoxia workstations in the 90s. And even Clean Air by Baker has been in the business since the 70s. So we truly have quite a bit of expertise in the field. But we do have the expertise, but we don't only manufacture these products. We have always been about sharing what we know. And over the years and decades we've been in the business, we have also, of course, gained invaluable understanding of the needs of science. And we have seen how science develops when research develops. So therefore, Baker has put in a lot of effort in supporting science. And we want to make sure that what we know is distributed to all of you who make that scientific work day to day. To that end, we support and host these webinars like today. We want to deliver science for science and support you guys. So without further ado, I hand over to Randall Johnson, Professor Randall Johnson. The stage is yours. Go ahead, Randall. Thank you.
Thanks very much, Krista and Daniel, and thanks everyone. And let's see, I'm just gonna, there we go. I'm just learning to use this particular webinar system, but I'm very grateful for the invitation, and hopefully we'll have an interesting time discussing this work of ours. So to start out with, of course, everybody sitting here in front of their computer screens is probably quite familiar with, you know, what hypoxia is. Otherwise, you wouldn't be sitting here. And I imagine because you're in this webinar, you're also aware of this particular issue, i.e. that tissue culture cells, as we commonly employ them, are cultured at a fairly high rate of oxygen, but really the level of oxygenation inside tissues and inside tumors in particular is much lower than that. And of course, cells are constantly going through different levels of oxygenation because they're constantly experiencing, depending on the tissue, different levels of perfusion and oxygen availability. And so any time in which the metabolic rate outstrips the availability of oxygen, we're in a state of what we might call hypoxia. But that definition of hypoxia means that, of course, what hypoxia is to one cell type and under one circumstance is different from what it is in others. So there's no real absolute definition of this. But the main take-home message of this little slide here is that essentially we know that hypoxic responses, we like to, as we describe it in this field and biologically, is probably something a bit less than room air, and yet almost all of our cells are exposed to that lower level of oxygenation throughout their entire existence. And so really hypoxic response is a misnomer, really. Hypoxia instead, or hypoxic response in a state of said response, is really something that cells are doing all the time under every circumstance. And I just have this little equation I like to torture students with on the left there, first propounded by an amazing physiologist, E. Newton Harvey. Really, he was working on clusters of small bacteria that would grow in spheres, but his description of how these needed oxygen on the exterior of their spheres to survive in the interior fits very well with things that we think about from the standpoint of human biology, including cancer and hypoxia and tumors. but I won't go into any real detail there because I'm not going to give anybody a test on that later. So let's see. So here's the sort of obligatory HIF slide. And I assume everybody here is quite familiar with that as well. So I won't really go through and spend much time on it other than to point out that, of course, a couple of years ago, this gave rise to a Nobel Prize. And that Nobel Prize was both for the discovery of the transcription factor itself, in this case, HIF alpha, but also for the mechanism for regulating that transcription factor. And of course, even for potential pharmacological mechanisms for modulating that regulation. Its regulation is, of course, elegantly through actual availability of oxygen intracellularly, the oxygen available to specific hydroxylases that covalently modify either prolines or an asparagine in HIF. Gives rise to either increased or decreased HIF function. And of course, this then becomes a mechanism for very dynamically and very immediately regulating response to oxygenation in a cell, and then ultimately how that cell can change its behavior transcriptionally to adapt to whatever level of oxygenation it happens to find itself in. And again, just a small number of definitions of terms. Really, the two HIF paralogs, HIF alpha paralogs that we're going to focus on are HIF1 alpha and HIF2 alpha, which are highly conserved, present in the vast majority of multicellular organisms, animals at any rate, and are the oxygen-sensitive components of the HIF transcription factor. So oxygen flux then is relevant to all the cell types in a tumor. And in the earliest stages of our analysis of this process, we, and I'd say the field itself, really focused fairly tightly on how hypoxia was relevant to the tumor cell itself, if we're thinking about tumor biology, and how that tumor-associated hypoxia likely drove a number of critical processes for cancer growth, including angiogenesis and the Warburg effect, modulating glycolytic increases in tumor growth. And it was only after, I would say, even in the last 15 years or so of hypoxia and HIF research that focus has shifted and pivoted to not just what cancer cells do when they become hypoxic, but rather how the other cells associated with tumors and even normal tissues change their behavior and adapt to differential levels of oxygenation, and then how in turn those shifts and changes and adaptations ultimately can themselves influence pathological processes, in particular and perhaps especially cancer. And this just shows in a cancer some of the cell types that we think about and have studied ourselves, and have of course been studied by many other groups in terms of hypoxic response, including monocytes and ultimately macrophages, peripheral T cells, and although I don't have them diagrammed here, endothelial cells themselves, all of which have very robust and different responses to differential oxygenation, and all of those responses differentially have been shown to influence tumor growth. And I guess our very first entree into this is just, I guess, quite graphically demonstrated by this slide, where you can see that in this experiment where we knocked HIF1 alpha out in macrophage and myeloid lineages more generally, we really removed a very significant component of inflammatory response when we just painted the skin of a shaved back of a mouse with a detergent solution. They get rather red because of disruption of barrier function and a subsequent inflammation. And then that redness is essentially almost completely prevented, and ultimately the inflammation prevented by loss of HIF1 alpha in the macrophage. So that argued for really the first time that HIF was a really important component of the immune system. So if we look at it in much more detail, we start to actually need to pick apart the various components of a tumor-associated response to by the immune system. And of course, we're now aware that there are inhibitory responses, probably regulated in part by CD4 Tregs as well as myeloid cells. In terms of immune response, there are cytotoxic responses, chiefly through cytotoxic T cells, but also NK cells. And then, of course, we have things like checkpoint blockade, the subject of the Nobel Prize prior to the one for HIF, that ultimately can impact on these processes and improve immunotherapeutic efficacy. And of course, what we've been thinking about for a few years now is how all of this is ultimately relevant to the manipulation of immunotherapeutic approaches via manipulation of the HIF pathway. And this shows just another sort of ancient paper from our group, which was one of our first just attempts to understand not just how any individual cell type acted in response to hypoxia and the immune system and tumor growth, but how interactions amongst cell types could influence the immune system's response to tumor growth. And what this showed in another knockout of macrophage HIF was that actually caused a slowing of tumor growth. So mice that had a mammary tumor model induced tumor growth when the myeloid cells lacked HIF-1 alpha. Ultimately, the tumors grew more slowly, and they also showed a decrease in tumorigenic progression. There was a much lower level of late stage carcinoma at a given stage of evaluation and much more precancerous tissue, indicating that we were not just slowing the growth, but slowing the progression of the tumor. And again, this is rather ancient data, but just to sort of point out that this really was a rather subtle effect. Ultimately, we didn't see much difference in cell death in the late stage carcinomas, but in the earliest stages, we saw a really significant increase in the amount of apoptosis. And essentially what Andrew Dodens, who was working on this problem at the time, showed in the lab was that we had a very significant increase in responsive T cells that could respond to by production of gamma interferon after a CD3, CD28 challenge to their T cell receptors in the myeloid HIF knockouts relative to wild types. And the way we interpreted that is we basically argued that in the tumor microenvironment, there was a suppressive microenvironment caused by wild type macrophages, and the loss of HIF actually alleviated some degree of that myelosuppression, allowing an increased level or a decreased level of energy or exhaustion in the T cells that had infiltrated those tumors. And that was a long time ago, but we just published a study to sort of continue that work. We've been working on this ever since, but we this paper I really would say was a very direct extension of what Andrew Dodens did. And this was Milos Gojkovic who did a study to basically ask to what extent, of course, do we see a suppression that, and to what extent is that directly related to HIF expression, particularly which HIF, HIF-1 or HIF-2, and I'll show you in a second, also related to HIF overexpression. And so what you can see here is that particularly actually under high oxygen conditions, but also under low oxygen conditions, when the macrophage are knocked out for HIF, and we do a T cell co-culture, where the macrophage have actually been pre-incubated with antigen that is presented to T cells that have specific receptors for that antigen. What we can see is in this antigen-induced proliferative response caused by the co-culture of these macrophages and these T cells, there is a hypoxic suppression of T cell proliferation in this co-culture, and in turn, that hypoxic suppression is primarily regulated by HIF-1 expression and not as much by HIF-2 expression. So arguing that there was a HIF-1 specific myelosuppression underway here, much in an in vitro system, much as we supposed was the case in vivo in that much older paper. And in fact, what we then were also able to see is that if we overexpressed HIF by simply knocking out the negative regulator of HIF, the VHL tumor suppressor, there, of course, as I'm sure are aware, you get very elevated levels of HIF because HIF is now being expressed in high oxygen conditions. And here you can see that in the context of the co-culture, there is a very significant suppression of T cell proliferation post antigen-mediated stimulation of or activation of the cells. And that's very clearly related ultimately to the increased expression of the VHL itself. Now, of relevance to sort of thinking about pharmacological manipulation of this pathway, we saw something quite interesting, which was that if we used a prolyl hydroxylase inhibitor, which is similar to some of those being employed already in the clinic to induce, for example, increased erythropoiesis, the FG4592. When we use this prolyl hydroxylase inhibitor, as you can see in the upper panel, this doesn't affect T cell proliferation much at all in T cells that have been induced to proliferate on their own. However, when we co-culture those again now with macrophages, the incubation with the HIF-inducing prolyl hydroxylase inhibitor causes a very profound suppression of T cell proliferation. Arguing again that it's this myelosuppression that is very HIF dependent and can be actually induced pharmacologically, that's acting to reduce T cell proliferation. And this is kind of I think an interesting thing to think about when thinking about the utility and potential effects of these drugs and manipulating this pathway. So what we show there is that HIF and myeloid cells can suppress cytotoxic T cells. We can show it in vivo. We can show it relates to co-culture experiments, and as such, we can actually induce it pharmacologically. But what about the T cells themselves? To what extent are they responding to hypoxia, and what does that hypoxic response do? And in work that's still unpublished, this just shows from Pedro Belisa in our group what oxygen actually does to a wide range of activation molecules. You can see in these are just FACS plots quantified, and you can see the numbers on the x-axis there are cell generation. So these are characterized by CFSE so that we can actually look at for each cell differentiation, or I'm sorry, cell division step post-activation, what the difference in expression is. And you can see under high levels of oxygen, we see significantly lower levels in many cases of activation markers and exhaustion markers like CTLA4 than we see with hypoxia. So low levels of oxygen actually have very potent and profound effects on a number of different aspects of cytotoxic T cell differentiation post-activation. Aziz Palazone in our group, when he was a postdoc, then decided to look at HIF-1 alpha knockouts specifically. HIF-1 alpha and HIF-2 alpha knockouts show quite different effects when those knockouts are specifically in cytotoxic T cells. But what you can see here is both that hypoxia triggers an increase in the expression, as that previous slide showed, of a number of markers of T cell activation in cytotoxic T cells, factors like gamma interferon, TNF alpha, and granzyme B. but that the expression of those is very significantly reduced when HIF-1 alpha is knocked out. The knockout of HIF-2 alpha is essentially has a much less significant effect, indicating that HIF-1 alpha is a potent trigger and a key factor in the activation of the cytotoxic T cell. And so if we think about that response in terms of actual effects in vivo, this is work that Andrew Dodens did when he was in Ananda Goldrath's group in San Diego. He looked in the Goldrath group at VHL knockout cytotoxic T cells, and what they saw was that VHL knockouts actually had reduced rates of tumor growth when in an adoptive cell transfer system, where he was actually able to look at an overall level of tumor control induced by specific tumor killing in these models. And what Aziz then was able to show is the converse was also true. Basically, in the upper left-hand side, you can see there that when you transfer in T cells specifically targeted to antigen expressing tumor cells, the T cells that are wild type in the dark round circles are able to control tumor growth rather successfully, whereas when those same T cells lack HIF-1 alpha, or T cells derived from littermates lack HIF-1 alpha, their capacity to control tumor growth is little better than animals that didn't get any T cells at all. So they basically have a very reduced capacity for cytotoxic T cell killing of tumors. And you can see the survival plot then to the right of that. And then if we looked at a checkpoint blockade experiment in the lower panels, you can see that there again, the loss of HIF-1 alpha in these T cells caused a very greatly reduced capacity to ultimately control tumor growth in checkpoint blockade, arguing again that the T cells themselves really have an absolute requirement for HIF expression in order for cytotoxic T cell function. And to sort of extend that, Pedro Velez, a postdoc in my group here at the Karolinska, had the idea of actually overexpressing HIF in T cells via retroviral vectors. And so what he did was construct a number of different retroviral vectors that differentially expressed mutant versions of either HIF-1 alpha or HIF-2 alpha for overexpression in T cells. And so because we knew that the VHL pathway was really important for differential function of T cells and obviously could give rise to increased HIF expression, we wondered what happens if it's combined with this other pathway, oxygen-mediated pathway of T cell, or I'm sorry, HIF control, the pathway involving the FIH gene. FIH modifies HIF transcriptional function. Also, as I said, oxygen in an oxygen-sensitive manner. And what Pedro did was actually ask, to what extent are these differentially acting the FIH pathway, which of course is a transcriptionally regulated or affects transcriptional regulation, versus the VHL and much better studied pathway, which acts through control of essentially HIF amounts present in the cell and the nucleus. And he took and actually produced a very large number of vectors, either containing wild type HIF-1 and HIF-2, or only HIF-1 or only HIF-2 there in the top row, or with mutations in the prolines there in the next row, that actually gave rise to their inability to be hydroxylated and thus became stabilized, or mutations in the asparagines that are hydroxylated by FIH, or mutations in all three sites that confer oxygen-mediated regulation. And they're then asked, okay, to what extent do these differentially affect tumor growth? And this was just published, but and I don't have time to go into a huge amount of detail here, but basically what he saw was that HIF-1 alpha, either in mutated or normal forms, had really sort of minimal effects, curiously, on increased cytotoxicity, but HIF-2 alpha overexpression gave a very potent level of cytotoxicity. And this just shows an in vivo cytotoxicity assay. But of course, we took that into animals, and we showed in a mouse system that we had a very differential an increased level of tumor killing and tumor suppression when HIF-2 alpha on its own was overexpressed. And what we also saw, quite interestingly, was that the effect of HIF-2 overexpression was essentially lost if the VHL sites were mutated. It was only when the FIH site was mutated that we saw a significant improvement in animal survival. And we took the mouse data and then decided to design a CAR T vector that we could use in human cells and employ in a human system to assay tumor growth in a lymphoma model placed where the lymphoma is placed into immunocompromised mice. And so using human donors that are transducted with either a CD19 CAR that reacts, of course, to the CD19 antigen on the tumor cells, or with a CD19 CAR plus HIF-2 vector, overexpression vector that actually has a specific mutation in this FIH site alone, we saw that we were able to get a very significant increase in control of lymphoma growth when overexpressing HIF-2, but only in this particular format where only one aspect of oxygen-mediated control had been eliminated. And what we were hoping to see is that oops, don't know what happened there. I guess we lost my, okay, I don't know what's going on. We don't have the slides anymore. I promise you that wasn't me. Let's see. Well, okay. Are we back on track there, Daniel?
Yeah, so I don't know why the slideshow went away, but you should be able to see it now, right?
Yeah. Okay.
Perfect.
I'll just keep going.
Perfect.
And so essentially what we're really quite interested in is then asking, can this have some clinical benefit? And that's something that we're in fact exploring right now. So I showed you that HIF can drive immunotherapeutic efficacy in cytotoxic T cells, that it increases T cell expression of effector molecules, that loss of VHL has been shown to increase anti-tumor efficacy. And of course, overexpression seems to do the same, although if you read the paper, you'll see that that's specific to a very particular effect on or mutation of HIF-2. And the last part of the talk, I wanted to talk about something a little bit different, but that is still relevant to this phenomena of tissue hypoxia. So this is a very old paper from our group, just showing that the so-called Pasteur effect, the increased level of lactate production during hypoxia, is essentially HIF dependent. And lactate, of course, is a key, an important metabolite in terms of thinking about the glycolytic aspect of hypoxic response. And because one of the most important inductions of lactate in normal life occurs during exercise and is as a result of skeletal muscle function, we were wondering about lactate's effect actually on T cells themselves. And this all came about because Helene Rundqvist, who's in my group now, has her own group here at the Karolinska, had been interested as a PhD student in exercise and its various physiological effects. And she decided in the group to try and extend that to ask, okay, to what extent does exercise-induced do exercise-induced changes influence T cell function, in particular T cell function during tumor formation. So the one thing that she showed first, and this is in a eLife paper that just came out a few months ago, was something that other groups have also shown, that in this model, but as I said, this has probably been shown in at least a couple of dozen papers now, from a wide range of groups, that exercise in mouse models can actually reduce tumor progression and growth. And she just shows this in animals that are able to run, as you can see in this way. They have a wheel in the cage and they're just able to run at will, which of course most of them do throughout the night, and we can measure the amount each individual animal runs. And in fact, the runners, compared to non-runners who have a wheel in their cage but it's locked so they can't run on it, the non-runners actually show a significant reduction in survival with the same tumors. And the thing that started getting us really interested in the relationship between CD8 T cells and running was the fact that when we did single cell isolations of spleen, lymph node, and tumor from runners and non-runners, the most striking difference we saw was that there was a very significant increase in cytotoxic T cells from the tumors of the runners compared to the non-runners. And in fact, we were able to do a depletion of the cytotoxic T cells in runners and non-runners. And
So for some reason, it seems like we lost Randall there. We're going to see if we can fix this problem for you. Maybe, Randall, you can close off and start again the video feed, perhaps. Try that, and I'll see what I can do on my end. Right, again, many apologies. For some reason, Randolph went off the platform, but we're trying to see if we can find him and get him to log in again.
hi, am I back?
Yes, you are, Randall.
Okay, good. I don't know
Did it
what
crash?
happened.
Or well, Nevertheless, you're back. Let's hope we don't have any more interruptions of that sort.
Yeah, it's not my internet because the internet is fine here, but okay. Anyway,
The wonders of technology, I guess.
yes, exactly. Okay, so to go back here, let's see. So anyway, yes. So if you use an isotype or use an anti-CD8 antibody to clear the cytotoxic T cells, you remove the exercise effect, and so that basically got us to start thinking much more carefully about what is exercise actually doing metabolically to T cells, particularly cytotoxic T cells. And one of the first places we went with this was to look at it from the standpoint of just mass spectrometric analysis of metabolites. And here just shows, not surprisingly, that these shift to a very significant extent when you exercise. This is skeletal muscle harvested immediately post exercise to exhaustion. Interestingly, there's a big downregulation compared to non-exercising animals of a number of the glycolytic intermediates, and a very significant upregulation of a lot of the oxidative metabolites. But more interestingly, we see differences between non-draining and draining lymph nodes, including a number of factors like 2-hydroxyglutarate and corticosterone that have potential immunomodulatory effects in the draining lymph nodes of skeletal muscle post-exercise. And we're continuing to explore the role of a number of these. And these changes can even be seen in plasma and, in fact, in spleen of exercising animals, arguing that there are actually a broad range of shifts in metabolites that are diffusing out into associated organs that have important immune functions. And it's not as simple as simply saying that exercise shifts the physiology of skeletal muscle, that in fact there are a range of effects extending themselves in all likelihood to the immune system itself. We wanted to test this in an in vivo system, so we carried out, Helen actually carried out this very ambitious experiment. She took animals that she could tag with a CD45.1 locus, allowed them to run voluntarily for 6 weeks, removed T cells from donors, either from non-running controls, animals running at a lower rate or animals running at a fast rate, and then put those into recipients, overall 60 different recipients, 5 per donor, to ask, do the T cells coming from exercised animals actually have an effect on tumor growth in non-exercising animals? So basically saying, can you separate the T cell mediated changes from the exercising animal and show that they are intrinsic to changes in the T cells themselves? And that, as you can imagine, was both a very ambitious and a very hard experiment to carry out, and in fact, even maybe a bit crazy, but it was exciting to do. And of course, we saw some interesting things. That's why I'm even showing it to you. If we looked at activation markers, here's one of the ones that was interesting. Of course, ICOS, which is one of the markers of an activated T cell. You can see that in the endogenous T cells from these non-exercising mice, the ones that they already had on the right, there were no real differences between those T cells from control or running mice. However, if we looked at lower or higher running rate mice, we saw significant changes between controls and running mice in the transferred CD8 positive T cells, are indicating that there really was a significant change in these T cells, even as they're in the animals that are themselves not running at all. And then when we looked at tumor growth, what we saw was it was subtle, but quite exciting. We saw a significant effect in the highest running animals in terms of tumor growth and survival between non-exercising and exercising. Now, it's not a big effect, but again, this is over a period of quite a few weeks, 6 to 7 weeks, that we're watching the tumor growth after these T cells have been removed from animals that were either running or not. So the fact that we see a persistent effect even weeks away from the exercise itself, argues that there's something quite interesting happening to C cells that are found inside running mice. One of the things that we know happens is that there's an increased amount of lactate in these mice. And one thing to remember about lactate, I think we're all sort of used to thinking about it as an immunosuppressive metabolite, but the lactate in tumors that is often focused on is, of course, lactic acid, whereas the lactate found in your blood is almost immediately buffered as soon as it's produced. And so we decided to focus in on sodium lactate as opposed to the anion, and ask, okay, at the kinds of levels induced by a high intensity exercise here in human subjects, levels approaching something between 15 and 20 millimolar, do you actually see shifts in T cell activation and function? And what Helen did was do daily sodium lactate injections to levels that will give rise to something between 15 and 22 or 15 and 20 millimolar spikes in the bloodstream for up to about a half hour to an hour as measured. We characterize this by injecting different doses to see what levels it would take to get up there. And of course, you have to put a fair amount of lactate into the animal. We went up to, in this experiment, 2 grams per kilogram. But what you can see is that, in fact, the higher doses of lactate actually had a significant effect on overall tumor growth, and in fact, increased survival and decreased growth to a statistically significant extent, quite unexpectedly. And in fact, what we saw in the tumors from those animals is there was an increased infiltration of CD8 positive T cells as well as CD4 positive T cells, and a significant decrease, interestingly, in NK cells during the course of those injections. So we were really changing infiltration parameters and profiles simply through a daily injection of lactate and a single spike per day, similar to what would have been seen from an intense bout of exercise. And so what I've had time to show you, even with these interruptions, was that oxygen-dependent physiology has profound effects on the immune system. It's a myelosuppressive effect when we're thinking about myeloid cells and T cells together. On the other hand, HIF response conversely is necessary for the action of cytotoxic T cells. We've shown that there are many tissue-specific roles for HIF in the immune response, and that these roles can be antagonistic in the context of thinking about things like tumor immunotherapy. And then in the end, I showed you that there was a very T cell specific role for exercise-induced reduction of tumor growth, and that manipulating lactate levels surprisingly appears to affect the role of T cells in terms of tumor growth. And the people in the lab currently include people who did some of this work, especially Pedro Lisa and Helene Rundqvist, Milos Gojkovic, where Milos and Helene have actually moved on already, and that we have a number of people continuing to work on these topics now in the other group I have, which is at the University of Cambridge. And then, of course, some of the people who used to be in the lab and did some of the work I presented early on include Andrew Dodens and Aziz Palassone. We're funded by a number of Swedish foundations, and as you can see there on the right, and in Cambridge by the Wellcome Trust, primarily. And thanks again for the opportunity, and I'm happy to take questions.
Yeah, thank you so much, Randall, for your presentation. And I apologize to everyone for those unexpected interruptions. Please write any questions you might have about Randall's wonderful lecture here in the chat pod down to the right. It's a chat box, and we'll bring them up on the screen. We do have one question already, so let me bring that up. So here we have a question by Hans Flery. Would you see a role for pharmacological reversal of the T cell suppression by myeloid-derived suppressor cells? My apologies, the pronunciation is a bit off there.
Yeah, that's something we're working on right now, and we even have a fair amount of data on it that we're in the process of writing up. So I can say yes. I can say there's a couple of interesting aspects of that also are currently unpublished. One of them is that unexpectedly, when we look more carefully and over a longer term, we are seeing now a role for HIF-2 alpha in cytotoxic T cell function. So that's something that we didn't expect. We were aware that HIF-2 alpha was probably one of the key players in myelosuppression, but it also appears to have an unexpected function in cytotoxic T cell anti-tumor activity as well. So that's been an interesting complicating factor. Something that I have that's still unpublished, but I've presented before this is when we knock out HIF-2 alpha in the myeloid lineage, we actually get an improved response to T cell mediated immunotherapies in a number of different models. So that certainly indicates that there could be an interesting answer to that question. But otherwise, I'm a bit constrained because the work's still in various places of evaluation. But certainly, yes, we could imagine that there might be a role for pharmacological treatment directed towards this myelosuppression.
All right, we have an additional question, which I will bring up to the screen right here, by Paola Casanello. How does age affect the modulary effect on HIF on immune function and tumor suppressive effect?
That's a really interesting question that I have nothing to say about, to be honest. Like most mouse researchers, we work on young mice, in large part because of how expensive old mice are. And so I don't, and we of course we do a lot of work these days on human cells, but not characterized by age because we get them from anonymized donors. So I don't have a good answer to that, although I could certainly imagine that HIF might well change during aging, and certainly the response to oxygen might change.
Thank you for that. And we have an additional question for you, Randall, by Helena. Did you test mitochondrial changes in your model? And anytime did you compare hypoxia versus hyperoxia?
We have been looking a lot at metabolic shifts in both T cells and myeloid cells, both actually after oxygen or after shifts in oxygen and after alterations in caused by exercise, actually. And we see some interesting things. In terms of general effects on oxygen or on relative to HIF in T cells, I can refer you to that Palazone et al. Cancer cell paper, because there's a pretty extensive evaluation by Seahorse and other means in that paper. And you can see that essentially some of the things we already know about HIF, like the glycolytic induction caused by T cell activation, is in fact HIF dependent. The hyperoxia versus hypoxia question, we have not done a lot on hyperoxia. I think hyperoxia is an interesting thing. Of course, it's not really, it's more of a treatment question, not a basic biology question, because I can't think of any circumstances in which animals normally experience hyperoxia. But of course, you can induce it. And it's a pretty good HIF suppressor, actually, in many regards. Of course, if you want to really go up, you have to start thinking about hyperbaric types of setups. And yeah, we have not explored that a lot. It's certainly an interesting question in this context, of course.
And thank you for that, Randall. If anyone else has a question, you're free to write that in the chat pod. We will see if someone starts to type any question.
I mean, the only other thing I would say about hyperoxia that we always think about, and of course everybody in the audience knows, I'm sure, is You only have to go up a little bit and you have to start worrying about explosions in the lab. I think it's anything over 30%, you've already got a hazardous situation. And the times we've thought about that, I have to say that's been a concern, both of us and the various safety officers in the institutions we're in.
All right. That seems to be the final question. Thank you so much, Randall, for taking your time to take part in this event. And
absolutely,
we're very happy and lucky to have you.
Thanks.
If there are no other questions, then I will take just 1 or 2 more minutes of your time to present our next presenter as well. That will talk about how oxygen can influence cell biology. Let me see if I can bring that up. Here we go. For our next episode in this webinar series, Sophie Molin will join us with her lecture, The Role of Oxygen and Hypoxia-Inducible Factors in Normal and Tumor Development. Sophie Molin is active at Lund University. She's an associate professor at pediatrics at LU and a personal investigator at the Lund University Cancer Center. We're excited to have her and hope to see some of you there. And of course, if any of you in the audience here are interested in holding a webinar highlighting the importance of oxygen in cell culture and cell research, please contact me at Daniel at milmedtek.se. Then I guess the only things left is to thank all of you attendees for attending to this webinar and allowing us to share this message with you. And we hope that we will see you the next time. So bye-bye. Thank you very much.